DNA is transcribed into messenger RNA, which is, everybody knows about messenger RNA nowadays because of COVID and the COVID vaccines being an mRNA vaccine, right? They change your genetics, which is pretty wild. So the mRNA travels to ribosomes. Ribosomes assemble amino acids into a chain called pre-pro-insulin. Ribosomes, endoplasmic reticulum, all those little things, those are organelles of a cell, right? A cell's made up of a bunch of organelles. They're like different parts of the manufacturing facility, right, inside of a cell. Pre-pro-insulin enters the endoplasmic reticulum. A signal peptide is clipped off and it becomes pro-insulin. So the pre is clipped off, it becomes pro-insulin. Insulin folds and bonds into proper 3D structure. So it folds and then it bonds with a disulfide bond. That's where DMSO, MSM, having those sulfur bonds are extremely important for insulin, and having cysteine amino acids, L-cysteine. This is crucial for function. Inside the Golgi apparatus, so the Golgi apparatus is another organelle of the cell, an internal segment is removed. That removed piece is a C-peptide. What remains is active insulin, two chains connected by a disulfide bond, right? Insulin is packed into vesicles and it sits there until blood glucose rises and it's released, right? The release trigger is glucose, right? Glucose enters the beta cells, it releases that trigger, ATP increases, calcium channels open up, and the vesicles fuse to the membrane. Insulin is released into the bloodstream, and so insulin, that's kind of how insulin is created, right? What it is 51 amino acids. It's a chain of 51 amino acids, and it's split up into two sections, kind of like this. You have an A and a B section. And what happens is when it's free flowing through the blood, it will link in to the cell A and B, and it opens up a channel for glucose to go into the cell to be created into ATP, more energy, right?